Okay, part 2 of the chapter 3 lecture. We were looking at flagella. This kind of bacterium called spirochetes. The CH is a hard sound, so spirochete. Spirochetes, when you look at them under the scope, they don't look like they have a flagellum, but it's actually because it wraps around the cell, and so you really can't see it. So we won't dwell on that too much. But they also have this interesting spiral shape, and spirochetes will be discussed later on because there are some spirochetes species that cause Lyme disease and syphilis. So there's some interesting spirochetes in terms of pathogens. But in terms of the flagellum, it wraps around, and it's that kind of flagellum is called an axial filament. But like I said, we won't dwell on that too much. For the typical type of flagella, in this picture, they're showing a peritrichous type of arrangement. But no matter what the arrangement is for the flagella actually turn like a propeller. So the part of the structure that goes through the cell wall and then the cell membrane is actually able to rotate in a spiral way, and it turns the whole flagellum in that way, and it makes like a propeller. And the reason I point that out is that in eukaryotes, the flagellum typically only goes like back and forth, not in a propeller motion. So it's a little different. But either way, flagella always help with locomotion. But with the bacterial flagella, the rotation, all of the flagella will rotate in the same way, and that causes them to all sort of bundle up. You can see in this picture here, and that's when the flagellum really gets some movement. And so when all the flagella are moving in the same, they're rotating in the same direction. That's called a run. And then when they start rotating in the opposite direction, it causes what we call the tumble. And the tumble is when the cell stops running. And then it starts moving all of them in the same direction again, and it does another run. So it's not the most efficient way to get from point A to point B, so to speak, because it's not typically, we don't think it's very direct, but ultimately the bacteria kind of eventually works its way to the attractant. But it has this sort of run and tumble, and it sort of goes in different ways and eventually works its way to the attractant, meaning whatever is causing it to be attracted to move in that direction. So another structure outside of the cell wall these are called fimbrie, and that's plural. These are little bristles, like bristle-like projections, but they're not flagella. In fact, they're shorter than flagella. In this picture, you have the fimbrie are these short, bristly-looking things, whereas the flagellum would be that really long one. And so a cell can have both. Really, the function of the fimbrie is to attach or adhere to other bacteria and to surfaces. When bacteria attach to surfaces, this is an important consideration because when bacteria attach to surfaces and to each other, they can form what's called a biofilm. And a biofilm is a growth of usually multiple species of bacteria on some kind of surface. And when they get in a certain proximity of each other, they start sending chemical signals to each other. And they can actually start to express genes that they normally wouldn't express because of this chemical signaling. And they can start to get more aggressive in like causing disease and so on. The most common biofilm that you're probably aware of is dental plaque. So the reason that you have to remove that every day is because as long as you keep removing it, it's never able to grow and get enough bacteria to become more aggressive. It doesn't start expressing those genes. So you have to keep removing it every day, and then we don't allow the biofilm to really progress to that point where it can really cause something pretty nasty in the mouth, for example. There are other biofilms that can form on anything that's inside your body. For example, a pacemaker can get a biofilm on it. Plates that are in your body that have been inserted in different places. A catheter that isn't cleaned properly can grow a biofilm over time. So there are medical items that can have these biofilms on them. So fimbrie would be one of the things that would help bacteria to attach and start forming that. Now, there's another structure. It's called peli. A pilus. Pilus is the singular, and then peli is the plural. Peli are these. They're really a special kind of fimbrie. They are these long extensions that connect two cells together, but specifically, they have they're like hollow in the center. So when two cells are connected by a pilus, the pilus will actually go through the cell wall and the cell membrane and create a connection, a direct connection between the cytosol of the two cells. And what happens is DNA can be transferred from one cell to another through that connection, through that tube. That process is called conjugation. So bacterial cells can pass DNA from one cell directly to another if they have a pilus. All right. And that's a special type of fimbrie. Now, part of our identification of an unknown bacterium would involve determining what kind of structures these cells have. So gram-positive cell wall, gram-negative cell wall, glycocalyx, fimbrie, pilus, flagellum. And also we look at the shape of the overall bacterium and whether or not there's any kind of arrangement in terms of other bacteria of the same type. And so on the left, you see a shape. It's a spherical cell shape that we call coccus. Coccus is singular. And listen to how I'm saying that, coccus. And then the plural is called cocci. Cocci is, the first C is a hard C, and the second C sounds more like an S. So cocci would be the plural. So in this image, you have one coccus here, but you have several cocci here. On the right side, you see a rod shape, and another name for that is bacillus or bacilli. Bacillus is singular, bacilli is plural. And those have that slightly elongated shape. And these can exist individually. We call those singles. So this would be like a single. But if you look on a slide where you've prepared a type of bacteria, you're not just going to have one, you're going to have a bunch. So what you're looking for is, do they naturally kind of form any kind of arrangement with each other? And if they're all kind of separated from each other, that would be singles. If some of them are grouping together, that little bunch, that's a cluster. And then I circled a chain. Typically, the cluster goes with a prefix called staph, and the chain goes with a prefix called strep. So you may see those prefixes as a hint. So streptococcus with a lowercase s would just mean spherical cells that are connected to each other to make these chains. If you had staphylococcus, it would describe spherical cells that group together in clusters. And if you have streptobacillus, that would be rod-shaped bacteria that like to form these chains. So there are also genus names that have these terms in them, but then you would see a capital letter indicating that's a genus name. So if it's a lowercase letter, it's just giving you the general shape and arrangement of the bacterium. If you see a capital letter, it would mean that that's the genus name of the species. Okay, so I mentioned a minute ago about the cell walls. So we need to talk a little bit about cell walls. There are fundamentally two types of cell walls. And the way we distinguish them is through something called the Gram stain procedure. And so we call the two types of cell walls, we end up calling them either gram positive or gram negative types. And what these are, all cell walls in bacteria are made of a substance called peptidoglycan. The glycan is going to refer to a specific type of carbohydrate. And the peptido is going to refer to some short polypeptide chains, some short peptides. So it's a combination of particular carbohydrates and particular peptides. All right. And what the cell walls do is they help protect the cell. They help to attach to other cells. They help protect the cell and give it its shape. I think I said protect twice. Okay, so what is peptidoglycan? All right, here's the word again. Now, all bacterial cells have a cell wall made of peptidoglycan. And remember, bacterial cells referring to the domain bacteria. Archaeans, that's another group of prokaryotes, they also have a cell wall, but it's not made of peptidoglycan. So only the members of domain bacteria have the peptidoglycan cell wall. And the carbohydrate portion, the part I said is hinted at with that root glycan, that carbohydrate portion is similar to glucose. So they put glucose on the left so you can compare. On the right, these are the carbohydrates that are in peptidoglycan. One is called S-acetylglucosamine, and one is called N-acetylglucosamine and N-acetylmuramic acid. Now, you'll be glad that we abbreviate these terms, NAG and NAM. But NAG and NAM are carbohydrates very closely related to glucose, but with some extra things attached on, little functional groups and such. You don't have to memorize the structure of NAG and NAM, but know that those are basically modified. They're carbohydrates from a modified glucose molecule. So you have these strands of NAG and NAM connected together. Like here in this picture, they just have one NAG connected to one NAM, but then there'd be another NAG and then another NAM. And this chain can go on and on and on. And then once you have one chain, imagine lots of chains stacked up together. And you start to get a sense of how the cell wall is constructed. So in this picture, the NAG and NAM are kind of coming at you in three-dimensional space. And you can see here's one strand, here's one strand. So all these strands are all stacked and bunched together to make and it continues down to make the cell wall. Then you have these little amino acid chains that are shown like this, these little amino acid chains shown like little beads, but those are amino acids in small chains that connect together these strands to each other. And so the sum of all of that is peptidoglycan, is the main part of the cell wall.